Okay, I'd like to show some material that we presented a couple of years ago at the AAPB. Some of the work we're doing using ZBuilder and sLORETA to image the changes in brain activation that go with photic, auditory, and pEMF stimulation. So everyone has always been interested in what are the changes in the EEG that occur when you stimulate. And this gives us a way to get a very good quantitative grasp of it. So what we do is ZBuilder is based on, as you may have heard earlier or will hear later, taking a reference EEG sample, could be a person, individual. It's a single sample, not really a database. And through ZBuilder, we can create a reference database that looks like a normative database in terms of the structure of all the numbers. It has means and standard deviations. But now when we analyze another piece of EEG, such as a test sample, using Brain Avatar, using analysis, the z-scores we get show the differences between the test EEG sample and the reference sample as z-scores. So we're not saying how normal or how typical the EEG is. We're simply asking in what ways is it similar or not to a previous EEG. Now, you can use this to look at changes in short periods of time, and that's exactly what we're going to talk about here. You take a discovery EEG, you need an echo or another stimulating device, stim to EEG interface cable so that we can capture the pulses. And you need Brain Avatar with, in this case, StimFlash software and ZBuilder, and you need a plan. So what we do is ZBuilder is something that comes up when you've selected EEG, and you can say, I want to analyze using ZBuilder all of the data or only the sections marked good, similarities or voxels, eyes closed, eyes open. And when you run ZBuilder, it creates a new reference. It is a database in a sense, but it's a database of one, N of one. So here's an example of some EEG we were using for this work. And as you can see, it's 19 channels of EEG with then these aux channels showing the appearance of the stim pulses. And on the last trace on the bottom, you see these pulses occurring, and they show that's when we either flash the light or activate the pulsed electromagnetic, which is the stim field using or an echo using micro Tesla coils, or headphones or earbuds, in this case earbuds. And so one might do evoked potentials with these by averaging the successive responses. What we're doing is actually easier. We simply measure the frequency content of all of the signals during a baseline and then compare it with the stimulation period. So the model we use, for example, here, we take 2 minutes of resting EEG, and that will be used in order to get the baseline. And the ZBuilder will build a data set from it. Then we do 30 seconds of 3.5 hertz stimulation. Then we rest for 10 seconds. Then we do 30 seconds of 7 hertz stimulation, rest for 10 seconds, 30 seconds of 14, and rest. And from a single trial now of this length, we get all the data giving us the changes due to these various stimulations. This is an example of the ZBuilder file, which contains all the raw data. This contains means and standard deviations for every channel, every frequency band, and as you can see, we defined custom frequency bands. With ZBuilder, you can use customized frequency bands. So we have a band centered at 3.5, one centered at 7, 10.5, etc. In addition to having delta, theta, alpha, low beta, beta, high beta, gamma. So we're taking full advantage. There are 16 bands available. We're going to use them all. Here's an example of the ZBuilder data for the voxels. For every one of the 6,239 voxels, we have a mean and a standard deviation in every frequency band, including the 3.5, 7, etc., etc. And these are now the reference. These are current source density values, so that, for example, on voxel number 1, the average value of the delta is this number, and the standard deviation is this number. And so it's showing, and these are actually logarithmic expressed so that they have all the proper qualities to be a reference database. And so we do a stimulation, and again, see on the bottom, you see all these stimulation pulses, and then they stop. And you can see in the raw EEG, you don't really see clearly the result. You might say, oh, I think I see more beta or something over here, but that's not a quantitative judgment. So this technique allows us to get quantitative information. Here's an example of what the report looks like. This is, we take this section, for example, 140 to 150 seconds. And this was a period during which there was stimulation. I think this was visual. And we then get z-scores. This is from analysis. And then these are for ROIs. We get ROI amplitudes as z-scores. You see a lot of z-scores between plus and minus 1, because it's rather difficult to get a large z-score when you do this kind of method, and we're publishing reports that show that. It's the repeatability and the reliability that gives us low z-scores generally, meaning things aren't changing. You need a z-score in the vicinity of 0.6 to 0.8 or greater for there to be a significant change. And you can see in some of these instances, we do have some significant changes. Here's another example, again showing negative z-scores show that there's a reduction. And generally what we find when we stimulate is there is a reduction in the endogenous frequency content of the EEG. Might be counterintuitive. You might think, oh, but we're stimulating and producing the rhythm. Well, the evoked potential itself is very tiny. It's just 1 or 2 microvolts. But more importantly, the brain stops producing energy at the frequencies when you stimulate repetitively at 7 and 10 and all that. So it's interesting. The overall energy in the EEG does tend to go down, but what remains tends to be phase locked with the stimulus. And some people think the evoked potential is nothing but the phase locking of the endogenous activity to the stimulus. Furtscheller, Klimisch, and others, and myself tend to move in that direction. Here's another report file. This is showing for the voxels. So for each voxel, we have z-scores for absolute power. And again, you can see generally these z-scores are small, between plus and minus 1. So again, even with these voxel values, it's when they significantly get outside or plus or minus 1, actually plus or minus 0.6 or so, when you have a significant change. And you see most of these are not significant at all, which is reassuring. That's what we expect. Here's an example, again, where we show the different frequency bands broken out. And we can rank then which parts of the brain have the greatest changes. And when you rank them using Excel, the ROIs pop to the top of the list. And for example, in this particular example, the frontal lobe, limbic, occipital, parietal show up according to that particular sorting. When we look at the results of stimulation, these are 3.5 hertz stimulation. We look at the effect of photic, of EMF, of combining EMF and photic, the effect of auditory, and the effect of the afterwards. Is there any lasting effect in the last 2 minutes? What we see is, not surprisingly, in the case of photic at 3.5 hertz, we see a reduction in delta, primarily in Brodmann 17, lingual gyrus, which is occipital, Brodmann 18, cuneus, occipital lobe, and the Hagman number 1, all visual system, the p-value here is less than 0.01. When we stimulate with EMF, we see the effects mostly in the frontal areas, 45, 46, 10, 44, even though the coils were actually over the motor strip. Interestingly, when we combined EMF and photic, we did not see any significant changes in the EEG. It was as if nothing was happening. When we did auditory stimulation, we saw changes in 90 regions of interest, very widespread reductions in delta, particularly the sublobar, the insula, 13 is the insula, of course, 33 and 24, temporoparietal types of areas, etc. Again, p-value of less than 0.01. Afterwards, the after baseline, no significant changes. In gamma, the photic stimulation at 3.5 hertz made no significant changes. The pEMF did, And in Brodmann 43, Hagman 3, transverse temporal gyrus, again, this is primarily temporal, temporal frontal, for frontotemporal. EMF and photic, no significant changes in gamma. With auditory, again, widespread changes in gamma. Again, the insula comes up, 44 comes up, which is dorsolateral cortex, frontal cortex. And again, at the end, there's no significant changes. So all these changes were found using ZBuilder in the way that I mentioned. Also, when you run analysis, you get maps. And you can move the maps and rotate them and change the scale. And here's the example of the 3.5 Hertz effect on delta, visual stim. You can see nicely localized to areas 17 and 18. Here's the effect of the pEMF stim, 3.5 Hertz on delta. Again, now we see these Brodmann areas, frontal 45, 11 kinds of things. Here's an effect of auditory stim. This is the insula. This is Brodmann area 13. These areas are beautifully targeted by this technique. Here's the auditory stim effect on gamma, showing the effect on the auditory system. Here, when you look at the top, you see we're actually getting networks. ZBuilder and analysis can pull out networks the same way it can with a normative database. Here's anterior cingulate, cuneus, very, very interesting. Here's the effect on delta from 3.5 Hertz. You see the left and right are very different. The left looks like this, again, an insula and some associated areas. And on the right side, you're getting a more diffuse temporal result. So you're actually able to see left and right changes right across that single session. So the findings we found is that all the stimulation modalities produced a decrease in the observed frequency bands. The effect of photic stimulation was localized to the visual cortex. The effect of pEMF was located primarily in the frontal areas. The effect of auditory stimulation was localized primarily in Brodmann areas 13, 33, 34, and produced specific network patterns in delta and gamma. The effect of combined photic and pEMF stimulation was significantly less than that with either modality alone, was not statistically significant. This is a very interesting new result. And during the at-rest condition after all stimulation, there were no significant changes. So another set of findings is the emergence of Hagman's hubs in certain instances. For example, photic affects Hagman 1, which is a visual system primarily. But the pEMF is seen to affect gamma in Hagman hub 3. Is the most significant effect. So because we're able to do z-scores not just for typical ROIs, but for hubs, you can find out if a hub is being affected by the stimulation. Or any other intervention. This technique could be used with hypnotherapy, any type EMDR, any type of intervention, high oxygen, hyperbaric oxygen. And one of the primary observed effects of the repetitive stimulation was to inhibit unrelated frequency activity in areas of the brain that are not related to sensory processing. So opened a lot of interesting doors, and we've learned a lot of interesting stuff. The effects observed were highly location and network specific. They were transient in nature. We view them as evidence of self-organization in which the locations and patterns of activation at particular frequencies exhibit specificity and selectivity. So the effects of stimulation are not well encapsulated in concepts like speeds things up or slows things down or relaxes the system, certainly not in training. We're looking at things very differently now. So we're better expressing things as dynamic, adaptive shifts in network, component activation, network interaction, as a response to an input that is minimal with respect to energy, but is more fully characterized with respect to information. So the concept here that frequency really is the main issue, not amplitude, and that we get different effects from different modalities. So I want to thank everyone for listening to this particular piece of the presentation. We think ZBuilder opens a lot of exciting doors. This is just one, and the results really bear out that the method is useful. Thanks.